today for this first talk in this series of lectures on insect-plant-pathogen interaction. These talks are part of the grad school program on the biology of the host-parasite interaction at the University of Goiás State in Brazil. And we'll have other talks here, another one tomorrow and two other on the 3rd and on the 5th of July as well. So you guys are invited as well. And today we have a special guest, Dr. Murad Ghanim, who is a senior scientist at the Volcanic Center in Israel and adjunct professor of entomology in the Hebrew University of Jerusalem. Murad graduated from the Hebrew University of Jerusalem and completed a 3-year postdoc at Yale University School of Medicine in the USA, after which he joined the Department of Entomology at the Volcanic Center. His research nowadays focuses on biological and molecular interactions between insect-vectors of plant pathogens, including whiteflies and psyllids, with plant pathogenic viruses and bacteria. He is also interested in understanding insect resistance to pesticides and the development of new and biorational pesticides with the industry and screening for active natural compounds. So Dr. Murad will talk for about 40 to 50 minutes, and then we will open the microphone for some questions, or you guys can also send us written questions in the chat, and then we can have some time for discussion as well. We thank Dr. Murad for being with us here today, sharing some of his knowledge and expertise. And Dr. Murad, please feel free to start whenever you're ready. Okay. Do you need help to share your screen?
Yes, I just click on the...
There's a square with an arrow pointing up, and then you click on that and share. You can share one screen or the whole screen, whenever you think is easier. Oh, also, I'm going to start to record here. I think it's a good idea to have this recording.
You see now the screen?
Yeah, I can see it.
You see it?
Yes.
Okay. So hello everyone. My name is Murad, and I'm from the Volcanic Center, as Patricia said, in Israel. And we basically focus on plant pathogen transmission by insects, and we focus on 2 pathosystems that we currently work on. One is a transmission of plant viruses by whiteflies, and we mainly focus on Bemisia tabaci whitefly, and which is a very important insect pest. As some of you know, it's important also in Brazil, and it transmits hundreds of virus species. In Israel, it has been very important in transmitting bigomo viruses, and the most important one was actually tomato yellow leaf curl virus, or TYLCV. And this is most of the works we did were with TYLCV. Recently, many other viruses came into Israel, and we also tried to work on some of them. And we usually basically focus on the interaction between the vector, the insect, and the pathogen, the virus. We do less work with plants. And we try to understand how those viruses are transmitted and what mechanisms inside the insect are involved in the transmission. Now, the other pathosystem we started working on about 10 years ago is the transmission of Liberibacter species by psyllids. And this system is currently present in Israel, is the transmission of Candidatus Liberibacter suranacearum, which is transmitted by the carrot psyllid, and causes a very important disease in carrots in Israel. Carrots are a very important crop and mostly goes to export to Europe. And the disease is called carrot yellowing, and it basically affects the carrot itself, and it's not marketable. And we try to collaborate also with people, scientists from the nowadays from the US, and there you probably heard about citrus greening, and it's also caused by another Liberibacter species and transmitted by another psyllid species. And basically this work started with this system in the US, and then we started working on the other system in Israel, and now we try to collaborate and look and see what parallels we can learn from both systems regarding the transmission of those bacteria by psyllid species. If I have enough time at the end, I will maybe talk a little bit about this system also.
Murad,
So yes.
We are seeing your notes there. Maybe you can just I don't know how to say that, or how can you take this off?
Oh, you mean you see the other?
Yeah. Maybe if you can just make it. Can someone explain how to do that for him? Because I don't know how to change this.
What do you mean the notes?
Below the next slide, maybe you can click no notes. I'm not sure, but.
Oh, you mean you see the next slide?
Yes, exactly.
Oh, I think there is a way to wait.
Because below the next slide there is no notes. Maybe if you click it there.
It's just because he didn't write anything.
Yeah.
Yeah. Yes.
Yes.
Good. Great.
Now?
Thanks, Mira.
Yes.
Okay. Sorry. Okay. So I basically I will focus on. I mean, I will talk about those systems, and I was planning to put focus on because we were using lots of imaging and microscopy in all of our studies, and I want to basically convince you that imaging and microscopy and using those techniques are not less important than using molecular biology and all the other techniques we know. We use all of the molecular biology as well, but I think involving microscopy and imaging and molecular probes and antibodies is basically, we think, is a much nicer and productive and nicer to the eyes and productive approach that can tell you many things that you don't see in other techniques. So we basically use light microscopy, transmission electron microscopy, scanning electron microscopy, and in recent years more and more confocal microscopy, and because we do lots of confocal microscopy, we also use many molecular probes, antibodies, and so on. So although in the title I say the imaging and localization of viruses, I will put those imaging techniques into context of the biological systems we work with. So I will start with this system of, as I say, transmission of tomato yellow leaf curl virus with the whiteflies. And you see in the bottom, this is actually the title of my PhD thesis that I did about more than 20 years ago. And it was titled Studying the Paths of Acquisition and Transmission of Tomato Yellow Leaf Curl Virus and How the Virus Affects the Insect. And because we needed to look at the paths of acquisition and transmission, we needed to do a deeper study on how those whiteflies compose from the inside. So we basically wanted to look at the internal anatomy of whiteflies. And this paper was the major paper we published that time, looking at the digestive and salivary and reproductive organs of the whitefly. And the conclusion of all this paper it's basically drawn here. When we look inside the whitefly, you can see the the head. The whiteflies usually feed from leaves through the stylet, and the viruses that are taken from the plant are acquired from the phloem by the stylets. They reach the gut, the midgut that you can see here. Patricia, you see my cursor also, or I need to use a pointer?
I can see the cursor. Yeah, I can see it now.
You see the laser?
It's better, the laser. Good.
You see it?
Yeah.
Okay. Okay. So so the idea was to draw the internal anatomy of the whitefly and then study how those viruses are transmitted by the insect, where those viruses reach and how they cross the digestive system, for example. And we are talking about TYLCV, the thermatoleucar virus, which is a circulative virus. And a circulative virus means that they are acquired, they reach the digestive system, they cross the digestive system to the hemolymph, they go back to the salivary glands, and from there they are secreted outside. And we usually use the aphids as our reference. Although aphids, if you look here, their digestive system is a bit different than whiteflies, because their digestive system is linear one. And you can see the foregut, the midgut, the hindgut, they are all one tube. And in the whiteflies, it's a bit different. So basically, one of the first things we started doing is doing sections through the whiteflies, and you can see here this red line, and this is basically and on the right you can see those sections, and this is basically a section showing the two pairs of salivary glands, the primary salivary glands in the whiteflies. This was the first time we actually could see organs that are involved in virus transmission by doing only sections through the whitefly. Next one is you see this line here, the red line, and a section through the thorax and the abdomen, and you can see how the esophagus is going through the thorax, then continuing to the midgut, entering the abdomen, and those were really neat, photos, and we're starting more and more, learning, and this is how we learned how the anatomy of this whiteflies is built from the inside. Now, later on, we started also dissections, and we said if we can do sectionings, we can do also dissections, and this is how the primary salivary gland look like in whiteflies. They are kidney shaped. And you see when we use different staining techniques, you can see that the glands, they do not stain uniformly across the glands. So you can see different regions here, and it turns out later that those different regions also have different functions. This is the midgut of the whitefly, and you can see here the same story with the staining. That when we stain, for example, with the toluidine blue, you can see that not all the midgut stains uniformly, and there are different regions, and also those different regions assign different functions in the gut, and also later on in the transmission. And basically, one of the most important areas we found in the gut is basically this area. It's called the filter chamber. And the filter chamber, we spent long time trying to understand how viruses basically arrive to the filtering chamber, where they are filtered, and how they continue across the gut, and where basically the food substances also that include the viruses are filtered across this organ, where the sugars are secreted, and how the viruses are absorbed to the hemolymph. So this is just to show you that many of these microscopy techniques help us to try and understand the structure of the internal anatomy of the whitefly, and how viruses can move across these organs and be transmitted. But then later on, when we needed more and more to understand virus movement and transmission inside the whitefly, we needed to develop more and more methods and techniques to see those viruses. And one of the first techniques, we surveyed a lot of the literature, and we tried to develop our own protocols for localization of these viruses. And here on the left, you can see fluorescence in situ hybridization of TYLCV inside the gut. And basically, when you look at this image, you directly see that most of the virus is basically localized to this area of the filter chamber. And this is why we thought that most of the absorption of the virus occurs in this area here. And you can see when you go along the midgut, the concentration of the virus is less and less. And this is using fish, and fish is basically a very simple technique of using very short DNA probes that are designed to hybridize with a small portion of the genome of the virus. In this case, is the V1 gene, which encodes the coat protein. On the right, you can see immunostaining. It's basically a similar technique, but using an antibody against the virus. And this antibody is also against the coat protein of the virus. So you can see that in both cases, you can very nicely see the virus, and you can see high amounts of the virus, and you can see also functional localization, because when most of the virus is localizing to the filter chamber, but we made this assumption that most of the virus is also absorbed to the hemolymph in this area. This is another image showing the immunostaining of TYLCV in the gut, mostly localizing to the filter chamber, and the lower panel is showing the localization in the primary salivary gland. And this is basically also made us think that this specific virus is basically transmitted through the primary glands and not the accessory glands, as for other viruses, for example, in aphids. And same techniques can be used also to localize. This is the localization of the same virus in tomatoes in the phloem, the sieve elements of the phloem. And you can see that localizes very nicely in the sieve element. And this is where basically whiteflies feed through their stylets, and this is where they acquire the virus for transmission. Now, those techniques were really powerful, and I'm not showing everything of these studies, but I'm focusing on those imaging and imaging of the organs, just to show you that those techniques are really powerful in understanding also what's happening during the transmission. This is a work that we published several years back, and showing the involvement of one protein that we identified using a microarray screen, and it's called heat shock protein 70. And we found that this protein basically interacts with the virus, with TYLCV, also in the filter chamber area. If you see the lower right panel, you can see that most of the co-localization, which is represented here with yellow, so the virus is localized with red. This is immunostaining as co-immunostaining of HSP70 and the virus, the TYLCV. And you can see in the lower panel that most of the co-localization, which is represented by the yellow color, happens here in the filter chamber. So again, we thought that HSP70 functions in this area here. And later on, we basically found also that HSP70 is part of the activation of the immune system against the presence of the virus. Because when we silenced or we inhibited the HSP70, there was more increase in the virus transmission. That means in a regular situation, HSP70 acts to prevent the virus maybe from being transmitted or being transported through the gut, because again, when we silence it, you get more transmission of the virus. This is another work where we identified another protein. is called cyclophilin. Cyclophilin was also shown to be involved in virus transmission in aphids. And in this case, with whiteflies, we also found that this protein was identified in a proteomic screen, and comparing infected and uninfected whiteflies. And again, you can see that cyclophilin also localizes. So you can see here a portion of the midgut, and the green dots that you see are cyclophilin. This is also immunostaining with antibodies. And in red, you see TYLCV, the virus. And when you do the overlap or the merge of the both of these photos, you can see that TYLCV mostly co-localizes with cyclophilin in the gut. And the same I think happens also in the salivary glands. So in the lower panel, you can see the salivary gland, the co-localization of both the virus and cyclophilin. And it's more interesting even because you can see that most of the co-localization happens here in the center of the gland. And this is, we know this is the area, this is an area called the secretory region of the gland. This is where most of the cells basically empty their content into the salivary duct. And the salivary duct goes to the stylet where saliva is basically secreted outside of the gland. So we know more or less that the virus interacts with cyclophilin in the gland. We don't know yet what exactly it functions. We know cyclophilins are chaperones that interact with many proteins. But we know they interact here. Again, if we silence cyclophilin, it affects very much the transmission of the virus. Now, now I want to move on to another aspect of the studies we did and how we also employed these techniques for understanding, and talk a little bit about endosymbionts in whiteflies. And endosymbionts are bacteria that live inside whiteflies, and they are basically widespread in many insect species. And they have diverse functions depending on which symbionts they are. This is the life cycle of the whitefly. And you see here the female that lays eggs usually on the lower side of the leaf. And those eggs, they usually hatch to a crawler, and the crawler develops through different nymphal stages. And you can see, if you look carefully on the nymphal stages, you can see those two yellow dots here in the nymph. And if we look inside these yellow dots, you can see that they are full with bacteria. Those dots are called bacteriosomes, and those bacteriosomes basically are transferred vertically from the mother to the offspring through the egg. Okay, and those bacteriosomes are full with bacteria, and this bacteria is called Portiera. Portiera is the primary symbiont of whiteflies, and primary symbionts are very essential for the species. They have to be there, otherwise the insects cannot live. But we distinguish between primary symbionts and secondary symbionts. So the primary symbionts usually complete the diet of their host, and they are very important for the species persistence. And now, because whiteflies and many other sap-sucking insects feed on the phloem, they feed mostly on sugars, and they lack many of the essential substances as amino acids, and those symbionts synthesize those amino acids for them. And that's why they are also housed strictly in those insects. Now, there are also secondary symbionts, which we know there are many species. They're found in many insect species, and many of them we don't really know what they do, what their functions are, but they were acquired through evolution within many insect species. In this photo here, you see this is an ovary dissected from a whitefly, a female, and you can see eggs in different developmental stages, and you can see those yellow dots here, yellow organs. And those yellow organs are basically the bacteriosomes that are acquired during the egg development. You can see those mature eggs, the big ones. They already have those symbionts inside. And you can see this one. This is immature egg that is now acquiring the bacteriosome inside. So the bacteriosome will go inside and the egg will develop and it will be laid later on. And this is how they basically take those symbionts with them and they can later on develop. Now in Israel, we did a survey and we looked on how those symbionts distributed among the two species we have in Israel of whiteflies. So we have two species. We used to call them B and Q species. Now they have different names. I will just use B and Q because it's easier. And you can see that they are not evenly distributed. For example, this symbiont, this bacteria called Hamiltonella, is found only in the B species. Wolbachia and Arsenophonus are found in the Q. Rickettsia is found in both species. And this is a survey done on individual whiteflies collected in different areas in Israel by PCR. Now, the interesting thing we wanted to see where those symbionts are located. And you can see by using also fish fluorescence in situ hybridization, using specific probes for each of those symbionts, you can see, for example, Hamiltonella is localized inside the bacteriosome with the primary symbiote. So in all these images, the primary symbiote I told you about, Portiera, is found in all whiteflies. So you will always see it. And it is localized here with the red color. Hamiltonella, it's a secondary symbiont. You can see that it's co-localizing with the primary symbiont. Arsenophonus, for example, is also co-localizing. So they are strictly found inside the bacteriosome. Wolbachia, for example, we found it's mostly inside the bacteriosome, but sometimes we can see some cells outside. But the only symbiont we saw it outside of the bacteriosome is Rickettsia. And Rickettsia is found in all the body cavity, but not inside the bacteriosomes. And this turns out to be a very also interesting bacteria that I will mention later. Now, as I said before, endosymbionts are vertically transmitted. That means they are transferred from the mother to the offspring. And as I told you, there are many bacteriosomes inside the hemolymph of the female. The bacteriosomes, this is an ovary also dissected from the hemolymph, and you can see here in red are the bacteriosomes after fish experiment where we localize the primary symbiont in red, and we localize Rickettsia is the secondary symbiont. Okay, so you can see those bacteriosomes. And you can see that Rickettsia is basically not inside the bacteriosome. It's found in the eggs, but not inside. So those bacteriosomes, as I told you, they enter the egg. So this is an egg reacted again with fish, and you can see how the bacteriosome is entering the egg. And later on, when the egg is laid, this bacteriosome moves basically from this area of the pedicel to the center of the egg. And when the egg hatches to the nymphal stage, this bacteriosome is basically divided into two bodies, two bacteriosomes. And later on in the adult, those bacteriosomes multiply more and more. And they swim in the hemolymph, and when they meet the eggs, the developing eggs, each cell of them will go into the egg, and this is how they are transferred to the next generation. As I said, Hamiltonella is this is a secondary symbiont. As you can see, it's Hamiltonella here is in green. The primary symbiont Portiera is in red, and you can see that is strictly co-localizing in the bacteriosome. And you can see nicely, even if you look very closely, you can see that Hamiltonella and Portiera, they don't occupy the same niche inside the bacteriosome. They have different areas where they are found inside this bacteriosome cell. This is another localization of Hamiltonella, and you can see very nicely how they localize inside, but they don't localize in the same areas. This is a localization in the abdomen of an adult female. You can see even the eggs here around. Now, as I mentioned before, when we did this survey of infection with those symbionts in Israel, we saw that Hamiltonella is basically infecting the B biotype, but not found in the Q biotype. This was really interesting because we know from other studies that symbionts might be also involved in virus transmission. And it has been shown also that one of the symbionts in aphids was responsible for efficient transmission of virus by the peach potato aphid. So we did really a large study that took several years with several students, and I'm showing you here just a conclusion of this study where we basically did we tried to determine if symbionts and whiteflies are involved in virus transmission, and which of those symbionts is really involved in transmission. And we found that Hamiltonella, this secondary symbiont, which I said is co-localizing with the primary symbiont in bacteriosomes, is basically involved in virus transmission, and it determines the efficient transmission of TYLCV by whiteflies in Israel. So when we did transmission experiments, you can see that all the B biotype or all the B species whiteflies can efficiently transmit the virus, while the Q not really good vectors for the virus. And those B biotype species, you can see they are infected. This one is infected with the Hamiltonella rickettsia. This one only with the Hamiltonella. The Q biotype populations are infected with Arsenophonus or Wolbachia, but no Hamiltonella in the Q biotype. So, and you can see we did later on many other experiments to show that basically a specific protein from this Hamiltonella interacts with the virus, and it aids the virus in its movement in the whitefly. So this is the whole story, and really very short. And if you are interested, you can go to this paper from 2010 and see really the whole thing we did. Now the model we work with in Israel, and we know it's also happens in reality, is that when whiteflies acquire TYLCV, it moves across the gut, it goes to the hemolymph, and in the hemolymph it interacts with this protein here that you see in green. This protein is called GROEL, and this interaction basically saves the virus from attacked by the immune system of the whiteflies. Because if we somehow inhibit this GROEL, the virus won't really survive in the hemolymph. So they move as a complex to the salivary glands. Here the virus is released and then enters the salivary gland, and then it is entering the salivary duct into the stylet, and this is how it is transferred to the next gland. And this really happens in reality. We know in Israel that when we have this is a greenhouse which has we can see tomatoes, and whitefly populations that were tested here were all B biotype, and this greenhouse was all infected with TYLCV after some time. On the contrary, if you see on the right, this greenhouse, for example, has eggplants and tomatoes, and we could see it was heavily treated with pesticides, and we found only Q biotype, and none of these tomato plants were infected with TYLCV. And you know, in Israel, if there are whiteflies, there should have this virus, unless there are a different. It's not B, only Q. Now we know that Q is also very resistant. So if there are pesticide sprays, then we have a Q biotype, but we don't have virus. So and this is really known in Israel. Wherever we have a virus epidemic of TYLCV, we know it's a B population. Now the other endosymbiont I showed you before is rickettsia, and I told you this was one of the interesting symbionts we work with, because it's not co-localizing with the primary symbiont in the in the bacteriosome. So you see here the bacteria. This is a fish again, fish experiment, where you can see the bacteriosomes in red, containing the Portiera, the primary symbiont, and in blue here you see Rickettsia. So you can see Rickettsia cells all over the body, but not inside. And this is a nymphal stage. Now, one of the interesting questions we really were interested in, since Rickettsia is not found in the bacteriosome, how it is transferred from one generation to another. Because if we maintain a whitefly population with Rickettsia, it will be infected, and Rickettsia will be transferred from one generation to another. But we know it's not inside the bacteriosome. So we did a study that was also published, and we basically followed the presence of Rickettsia using fish, using other transmission electron microscopy techniques, and we could basically show that Rickettsia infects the eggs during development. So Rickettsia is found in the hemolymph. It enters the eggs that you can see here. So Rickettsia here is labeled with green, and you can see even at the level of the single cell of Rickettsia, they are found everywhere in the ovaries. And when the ovary develops, Rickettsia cells enter the cells, and this is how they are basically infecting the next generation. This is called the transovarial transmission of this symbiont, but it's vertical transmission, but it's not through the bacteriosome. It's by infecting the developing ovaries inside the female. Now, one of the really interesting observation we made lately is, so here you can see with Rickettsia, here you can see two images of Rickettsia localization in the female, in the whitefly adults, and on the right you can see Rickettsia infecting the whole body cavity of the whitefly. So in red you see the primary symbiont, and in blue you see Rickettsia. But on the left, the difference is that this whitefly is infected with TYLCV. So the difference here, that you can see that the uninfected whitefly is infected with Rickettsia all over. And the infected whitefly with TYLCV, Rickettsia is somehow concentrating around the bacteriosomes. And we don't really have a good explanation why this happens, but we can see from other images, you can see here the same thing. This is an infected whitefly with Rickettsia, and you can see all those bacteriosomes in red with the Portiera, and you can see Rickettsia in blue, and you can see how Rickettsia cells are really concentrating around the bacteriosome and trying to somehow maybe enter the bacteriosome. We don't know. But we can see the same thing here. You can see it very nicely. And here, this is 10 images, and you can see how many Rickettsia cells are around the bacteriosome here. Inside the bacteriosome, and this is a V biotype, and it has Portiera. Those are Portiera cells, and Hamiltonella cells inside the bacteriosome, and Rickettsia cells around. So we don't know why Rickettsia cells are really, when the whitefly is infected with TYLCV, are concentrating around the bacteriosome, but this is a phenomena that we always see in infected whiteflies. Now, as I told you before, Rickettsia, when we started this work, Rickettsia infects both whitefly species, B and Q, but you see that Rickettsia is not infecting 100% of the individuals. So there are some individuals that are not infected with Rickettsia. And this was good for us because we could establish populations infected with Rickettsia and populations that were not infected with Rickettsia. We basically take individual whiteflies, we give them to lay eggs, then we test if they are infected or not, then we combine all the infected and all the non-infected ones. And this is how we established Rickettsia infected and uninfected populations. And those are basically the populations we use to see how Rickettsia can affect the whitefly. And we really tested many biological parameters, and we saw that rickettsia have different effects on the biology and development, on even on pesticide resistance. So there is a response to pesticides. So if you have whitefly populations in the field, one of the nice things to see or to test if they are infected with those symbionts or not. So this is the infected population. This is the uninfected population, and you can see a big difference in the amounts of rickettsia that are found everywhere in the body. Now, if we look at the gut of the whitefly, and we see that rickettsia is also infecting very heavily the gut. So this is a gut with the fish experiment localizing rickettsia. So this is using a specific probe for rickettsia, and you can see that all this red are rickettsia cells. So rickettsia is found everywhere in all gut cells. And if you remember this gut I showed you before, this is infected with or this is after TYLCV acquisition. And one of the questions we really asked if whether there is any interaction between rickettsia and TYLCV in the gut. Because if they both found in the same cell, there might be, any kind of interaction happening there. And here, when we did many fish experiments looking at rickettsia and TYLCV in guts, one of the things that we noticed is whenever you have a heavy infection with rickettsia, you have less virus in the gut. So that here you see two guts. One is rickettsia infected and one is rickettsia uninfected. And here we are localizing the virus. So TYLCV is in red. So this one is infected with the virus, but not infected with rickettsia. And this one is infected with rickettsia, but no virus. Okay. And you can see here this is the same image, but now showing rickettsia. So you can see high amounts of rickettsia, less virus in the gut, and here you see high amounts of the virus, less rickettsia, okay, or no rickettsia. So, one of the conclusions was that there is some antagonistic relationships between rickettsia and TYLCV in the gut. Now, the other interesting things we see, and we're still working on this issue of rickettsia and involvement in virus transmission. If we compare rickettsia uninfected and rickettsia infected populations and retention of the virus, you can see that when we have rickettsia in the whitefly, there is better retention of the virus in the. So this is again fish experiment showing TYLCV and showing rickettsia. So when we have rickettsia, we have more virus inside the whitefly, okay? And when we don't have rickettsia, we have less virus. So this is not as the previous experiment I showed you here, just looking at retention, okay? And regarding retention, we know if we do retention experiment where we do acquisition, then we look at the amounts of the virus in the whiteflies, you can see that, and this is a time course, 24 hours, 6 days, 2 weeks, and 3 weeks. You can see that in the rickettsia uninfected whiteflies, the virus is retained less in the insect. So the presence of rickettsia has to do something with the virus retention in the whitefly. And this is also can be seen in the transmission experiment. When we do transmission with rickettsia infected and uninfected whiteflies, you can see that the rickettsia infected ones are better transmitter or better vectors for the virus than the rickettsia uninfected. Is almost double the amount of transmission by rickettsia infected. Again, this has a very important implication in the field. If a population is infected with rickettsia, it will be a better population in transmitting the virus. And the last thing I want to show you regarding rickettsia, this is not related much to the previous one, but one of the observations we made when we looked at rickettsia and the whitefly is that rickettsia was also found. So those are the two pairs of the pair of the primary salivary glands from the whitefly. And when we did the fish experiment, we could see also rickettsia inside the glands. So if rickettsia is found inside the gland, then one of the ideas was to test whether rickettsia is also secreted into the plant. And this was really the case. If we do a fish experiment using rickettsia probes inside the plant, you can see that it's found in the phloem. So rickettsia is basically entering the glands and then secreted into the plant when the whiteflies are feeding on the plant. This is rickettsia localization in the stylet of the whitefly. Now we don't think rickettsia infects the plant. We know that it is injected into the plant, but we know that it's not surviving much into the plant. So it's not a plant pathogen. Is an endosymbiont in whitefly, but it is injected into the plant when the whitefly is feeding from the phloem. We don't know if it has any effects on the plant. We never tested that, but you can see that it's really found also in the phloem. So just to summarize this work with whiteflies, we really started from looking on whitefly anatomy, and then we came through looking at the endosymbionts in whiteflies, looking for different proteins. I showed you 2 examples of proteins that are involved in virus transmission, but we know this is more or less an updated image of what's happening regarding virus transmission. And today we know, those works were done by us and others, and today we know enough number of proteins that are involved in virus transmission. Some of them were even targeted by different approaches, and showed that those proteins are important in virus transmission. And the idea is how we can employ this knowledge in interfering with the transmission. Patricia, should I stop here? Because the other part is related to rickettsia. And I think we used the time, right?
Time is okay. I don't know about your time, but
no, my time is fine. I just want to be sure I'm okay with time.
You are. Please go ahead.
Okay, so I will try to do it quick with psyllids. I just wanted to show the other system we work with, and this system has been we've been working with this about 8 or 9 years ago now. And as I mentioned at the beginning, in Israel we have this carrot yellowing disease, and it's caused by Liberibacter solanacearum, and it is a bacterium. It's a fastidious bacterium that we cannot grow really outside of the plant or the psyllid, but it's transmitted by the carrot psyllid, and it causes this really very important disease in Israel, and it's found all over Europe and many other countries. But the most important disease caused by other Liberibacter species is citrus greening. I know in Brazil is important, in China, in the US, really very important. And this other Liberibacter species, Liberibacter asiaticus, is transmitted by the Asian citrus psyllid. By the way, it was discovered about a year ago in Israel, but without the bacteria now. So we might be able also to start working with this. But our interest in this system came because we are interested in circulative transmission of pathogens. So this is the Asian citrus psyllid, and this is more or less the internal anatomy. And you can see again that those bacteria are phloem limited, and they are taken by the psyllid to the gut. So the gut looks like this in psyllid, and there is a hind gut, and the bacteria reaches the gut and it crosses to the hemolymph, goes back to the salivary glands, and this is how. And then from the salivary glands they are injected into the plant phloem. This is a fish image showing the localization of Liberibacter in the phloem. Now, one of the things, the first things we start doing when we looked at this image looked at this system is basically how Liberibacter is localizing in the gut. And when we did those first experiments, you can see that. So Liberibacter is in red, and this is Liberibacter asiaticus that causes the citrus greening. This work was done in Michel Celia's lab in Cornell University, and you can see that Liberibacter is really colonizing the whole gut. But one of the interesting things we noticed that the gut doesn't look really normal, and the structure the structure is really weird. And when we started looking at guts, just by structure, not even localizing the Liberibacter, you can see the difference. So on the right, you can see a gut infected with Liberibacter, and on the left, you got not infected with Liberibacter. And you can see the difference. And you wonder how those psyllids can survive with such a really messed gut. So on one hand, Liberibacter wants to be transmitted to other plants, but on the other hand, we don't think Liberibacter wants to kill the psyllid. So there is really a cross talk between, there is a kind of a control of Liberibacter of what's going on in the gut. And we assume that also the psyllid is counteracting against this infection in its gut. So the other observations, what you can see in those, so here the nuclei are in blue. They are stained with dapine. You can see how the nuclei are really disintegrating when the gut is infected with the bacterium. And this was an indication of that there is a cell death or programmed cell death is going on in the gut. So it seems that the psyllid is activating immune responses against the bacterium. And I remind you, this is how the healthy gut or an uninfected gut looks like. So you can see the difference in the, and also you can see the differences if you do transmission electron microscopy. You can see on the left, this is a normal nuclei, and see how nice is it. But on the right, you can see this one is infected with Celas asiaticus that causes the greening. This one is a gut of the carrot psyllid in Israel infected with the other Liberibacter species, and also causes effects on the gut cells. But one of the really interesting, and this is what really afterwards drove us to do many other things with those systems, is how when we did those sections or EM sections, we could see that the ER, the endoplasmic reticulum in the gut cells, is really forming very nice structured, we call them later vacuoles. And it turns out that those vacuoles are induced after Liberibacter infection. As you can see here, they are causing, and we call them Liberibacter containing vacuoles, because later on when we did the more sections, you can see that those membranes are formed by the ER, the endoplasmic reticulum, and if you look inside, you can see those Liberibacter cells inside. So we think, and this is not new, this is known from other bacterial, even human bacterial species that cause different diseases, that they use the ER as a site where they can replicate. So they basically use the ER to build these vacuoles and then replicate inside. And again, you can see here these vacuoles. This is ER tracker, which is a marker that we can stain ER, and you can see these vacuoles are here, and inside you can see Liberibacter. And this is in both systems, the Celas, the greening system, and the carrot system in Israel. In both cases, we can see vacuoles formed. Now, thinking about the ER is one of the most really interesting organelles found in the cell. And in the ER, you know that proteins are synthesized there. But the ER is really one of the very active organelle in the cell. And whenever there is stress on the cell, there is also ER stress. And if there is a ER stress, the cell will always respond depending on the kind of stress. And you can see that. So this is the ER site, the ER lumen here, and this is the cellular cytosol here. So you can see when there is a ER stress, there are different responses activated. Depends if whether this is a heat stress or whether there is a viral infection or bacterial infection, and one of those pathways will be activated or more to respond to this stress. And it turns out also that Liberibacter is really involved in the activation of some of these pathways. So we have different projects now looking at, for example, calcium signaling. This is a work that was published recently. We have another work that looking at the ERAD. The ERAD is the ER associated degradation. And this is a mechanism by which unfolded proteins as a response of the stress are taken from the ER outside to the cytosol, and they are degraded there by ubiquitation. So we know that Liberibacter interacts with a major protein here called darling, and we think that this mechanism is activated to basically shuttle outside maybe bacterial proteins and degrade them, and this is a kind of immune response. And I have also another student working on the UPR. It's an unfolded protein response. This is a response. This is another response to the infection with the Liberibacter. And we use here really different techniques and approaches and methods. You can see here some, we use many ER stress inducers, and we use many inhibitors of some of the proteins you see here. And you can see all of these arrows here. So we look at the expression of the genes here. We know that autophagy is involved. Autophagy is also kind of an immune response. So we basically, our focus now is on ER and how ER is involved in the response to Liberibacter. And we really now understand that Liberibacter is orchestrating the or trying to control the responses that happens here in the ER, not to lead the cell to apoptosis and death, but to maintain the cell for its own replication and then later on transmission. This is just an example. I won't really put time explaining, but this is the darling, this protein from the ERAD, the ER-associated degradation. And you can see that this protein, when the psyllid is infected with the Liberibacter, you can see that it is induced. So this is the localization of the bacteria. This is localization of the protein. So those approaches can also be used. Those fish and immunolocalization approaches can be used to localize also candidate proteins that you are interested in. And when we do co-localization with the bacteria with the so this is darling, this is a bacteria, and this is the co-localization. You can see that darling is very nicely co-localizing in the gut in this stripe-like pattern. And when we do silencing of darling, you can see that the co-localization is completely abolished, and this affects very much also the transmission of the bacteria. So we know that ERAD is an important mechanism that responds, and if we silence a key protein in this response, we also affect the transmission. And we know, I told you that one of the responses is apoptosis, and you can see here those guts dissected from the carrot psyllid, and you can see that this blackening of cells or nuclei. This is one of the responses we always see when the guts are infected with Liberibacter. So this is also to visualize real effect on the guts. And lastly, I just want to mention that we do many other studies on these interactions. These two studies that I'm showing here, they were published lately. If you are interested more, so this is the immune response and the involvement of autophagy in the response to Liberibacter. And again, we use all these different imaging and localization techniques to show that the bacterium is inducing immune responses and inducing lysosomes, inducing autophagy in the gut to protect the gut from the bacterial infection. And the last example is we also showed that the actin cytoskeleton is basically a very important component of the cell that the bacteria uses to move inside the cell during the transmission. You can see here, this is an important protein in the actin cytoskeletal filament inside the cell. And if we silence this protein, you can see, so this is a control where you can see how the bacteria is nicely co-localizing with the actin. But if we silence this gene, you can see that the co-localization is completely affected, and this also affects the transmission of the bacterium. So the different mechanism, different ways to look at, and also again using those imaging. You can see that almost every experiment we do, we try to use those imaging methods, and they are always nicer than doing PCR or other molecular ways. Now, I just, if I summarize, I just wanted to put focus on how you can employ and use imaging and localization techniques, and recent years, and maybe the last 20 years, there is an explosion of methods and that you can use with this regard. Different molecular probes, almost every gene now or protein you want to work with, there are antibodies, and antibodies that cross-react even from different species with your species. And it has, I think people now are recognizing more and more those methods. And you always need to look and do microscopy before doing many other things. And all the things I described were done by many people in my lab, in other labs, collaborators. I don't have all the names, but I try to cite all the papers that were published. And the funding comes from different sources in Israel and local in Israel, or international collaborations with usually there are binational collaboration with other countries. And thank you. I hope I was not too long. Thank you for your listening. And if you have any questions, I'll be happy to answer.
Thank you so much, Murad. It was amazing, really nice presentation. We already have some questions for you here in the chat, and I also have someone who wants to do in the microphone, I guess. So I'm gonna go in the order here. So Simone asks, TYLCV circulates inside the whitefly. So is it possible to localize non-circulative viruses in whiteflies and aphids as well using these techniques, I guess is her question.
Yes, sure. It has been done with with non-persistent viruses, with the semi-persistent viruses, and we actually work now on semi-persistent virus transmitted by whiteflies. It's in sweet potato, and those techniques and fish and immunostaining. If you have good antibodies, you can and those things have been done also by other groups and in other systems. So it definitely can be done, and you get very nice, very nice. Usually the non-persistent or semi-persistent viruses are will localize to the stylet or the foregut, but they can be done also. I mean, it's been done in several systems.
Cool. So I thought someone asked to talk in the microphone, but it's not here anymore. But we have some other questions in the chat. So Marcos asked, thank you for the excellent presentation, Dr. Murad. In the Bemisa-Tabasi model, is it possible that TYLCV uses the Rickettsia bacterial cell for replication?
Uses the Rickettsia for replication? That's the question?
If TYLCV uses Rickettsia cell for replication?
Um, no, I don't think so. TYLCV has been shown in just in one case that it accumulates in whiteflies. I mean, kind of replicates, but this is now the accepted hypothesis is now those bigomoviruses to which TYLCV belongs also they do not replicate. I mean, there is only one case which we showed that if you impose only a stress on the whitefly, the virus can be accumulate a little bit, but this is not. We don't think it's a really. You can call it replication, but it's not. We don't think rickettsia is related. We don't think there is direct interaction between the virus and rickettsia. We never saw direct interaction. I think if there are any interaction, they are indirect through the whitefly cell.
Okay. Murad, can you stop your presentation so that we can see your camera better?
Stop.
Yeah, there's a bar.
Stop sharing.
Yeah. Yes, good. So one more question here. Pedro Gonzalez asks, any evidence of ER stress produced by plant viruses and their vectors?
Plant viruses.
Yeah. ER stress, he asks.
Yeah, I'm not sure I have ever seen such an example. But it doesn't mean that it doesn't exist.
Yeah,
there
might
be some examples. But from this TYLCV system, we know that TYLCV induces many responses in the whitefly. But I don't think any of those studies have addressed the involvement of ER. And usually, they looked at many autophagy was shown to be responsive to bigomoviruses. But I don't think ER was implicated.
All right. Any questions, guys? Any more questions here? Someone wants to talk in the microphone or something? While you guys decide about that, I just want to thank Murad again, and and share with everyone here that Murad is coming to Brazil in September. He's going to spend some days in the Inseto-Planta-Patógeno, in Brapa in September, and then after that, he's going to give a presentation in the Brazilian virology meeting. It's going to be very nice to have you here, Murad. We have a lot of things to talk about and work together, right? So, if you guys don't have any more questions, anyone? Last chance? Okay, so we thank Murad again. Very nice presentation.
Thank you very much. Thank you for everyone being here.
Yeah, and we will see you in September.
Yeah, thank you.
Around here. Thank you so much. Nice, guys. Thank you.
Bye-bye. Thank you.
Please sign the attendance list. Who haven't yet?
I leave, okay?
Oh, what?
I can leave? Or...
Yes, you're okay. Thank you so much,
okay.
Murad. Yeah.
Thank you.
Bye-bye. Thank you, Murad.
Thank you. Bye.